Welcome to this quarterly webinar hosted by Cure Parkinson's and the Journal of Parkinson's Disease. My name is Thilo Kunath. I'm a professor at the University of Edinburgh. And today we're going to hear about clinical trials targeting alpha-synuclein. So since 1997, alpha-synuclein has hit the research scene as a target for Parkinson's disease because of genetics and because of pathology. And today we have three brilliant guests that are going to give their perspective on this topic. So at the bottom of your screen, if you have any questions, please put them into the Q&A. And we're going to try to get to as many of those questions as we can during the webinar. But any questions that aren't addressed during this talk, the team at Cure Parkinson's will try to answer them either directly or compile them to get answers to you through frequently asked questions. Okay, so my first panelist, I'm just going to introduce them one at a time, is Hilal Lashwal from Lausanne. He's the director of the Laboratory of Chemical Biology and Neurodegeneration. And I'm going to let him introduce himself. Please, Hilal.
Thank you very much. It's a great pleasure to be with you all today. So my name is Hilal Lashwal. By training an organic chemist, biochemist, practicing neuroscience without a license. I've been working on synuclein for almost more than 20 years. My interest in synuclein started with trying to decipher the mechanism of synuclein aggregation with an emphasis on identifying the different intermediates on the pathway to fibril formation. We later then turned our attention to trying to understand the role of post-translational modifications in regulating alpha-synuclein aggregation, seeding, and pathology formation. And this was driven by the fact that the majority of synuclein and pathological aggregates are modified, and we wanted to decipher which of these modifications are responsible for driving pathology, which ones represent a response to pathology. And over the years, we realized that working in a test tube and the reductionist approach has its limits. So we moved on to try to understand the mechanism of synuclein aggregation and pathology formation in neuronal models, with the goal of trying to develop neuronal models that recapitulate the processes and the pathology as it appears in human. And for this, we use quite often the seeding alpha-synuclein models. As we began to understand how synuclein aggregates in cells and the complexity of synuclein aggregates, we realized that we needed to go back to basics. And therefore, that is to understand synuclein pathology as it happens in the brain and use the information we get from that to improve our models and our hypotheses. So in the context of this, we have also developed a number of tools and methods, including antibodies and mass spec-based methods, to remap, quantitatively map synuclein PTMs in the brain, and to try to study synuclein pathology in the brain. As some of you may know, my lab is also actively involved in conducting a number of sort of development of methods and number of replication studies in this field. And I think I'll stop here. Again, thanks for the opportunity to be with you today.
Great. Thank you, Hilal. So our second panelist is Gennaro Pagano, from a group leader at Roche Pharma Research, and he was the lead researcher for the Pasadena monoclonal antibody trial. So Gennaro, your introduction, please.
Thank you for inviting me. It's a pleasure to speak here today, and I can give a bit of myself. I describe myself as I'm a doctor, I'm a neuroscientist, I'm a drug developer, and I'm a dream builder. As a doctor, I studied the brain, right? I'm a specialized in neurology and work a lot with Parkinson's disease and Alzheimer's disease. And as a neuroscientist, I focus on trying to understand what's happening in the early stage of this part of this disease, right? What is driving the progression of the disease, starting on the genetic models, and in particular, I had the opportunity to work with the synuclein carriers of F53 mutation, among other different genetics. But this was really, really important to understand what are the drivers that could be potential targets of drug. And of course, by working also in academia, mostly in the UK, also done several clinical trials in collaboration with industry and academic studies. And then I decided to join industry approximately 5 years ago and I joined Roche that decided to invest heavily on Parkinson's disease. And my first program, and you mentioned before, was prasinezumab. And I'm proud to say that over the last 5 years, we really invested heavily in Parkinson's disease. I'm also leading another program that works on inflammation, but we have several research and we expanded significantly our team. We recently, about a year ago, we also got Patrick Brundin joining as a therapeutic area leader for the movement disorder. So we're really focusing now to try to stop Parkinson's disease. And I say I'm a dream builder. I stop when Parkinson does, is my motto. And I think that we are creating a future with drugs that might work. Hopefully some of them will, or at least one of them will, that can create a future where people with devastating brain disorder can be in control of their life. And this is why I'm here, right? And thank you for inviting me.
Brilliant. Dream builder. I love it. Thank you. A third excellent panelist is Carol Siu. She is a person living with Parkinson's. She's also an advisor to UCB Pharma, who have a strong interest in alpha-synuclein and Parkinson's disease. Carol, please.
Good afternoon, everyone. My name is Carol Siu. First, I would like to start by thanking Cure Parkinson's for inviting me to this webinar discussion. I was diagnosed with Parkinson's disease 7 years ago. I trained as a nurse back in the 1970s. For the last 25 years of my career, I was a university lecturer teaching research methodologies to healthcare professionals. I maintain my interest in research designs, particularly using mixed methods. In other words, using both quantitative and qualitative designs to complement each other. And I jointly published a book on this type of research method in 2013. Since my diagnosis, I work closely with Parkinson's UK charity as a patient volunteer. I had been a member on both Research Support Network and patient and public involvement steering groups. At present, I focus on the role as a chair of the South East of England Parkinson's Research Interest Group. Our aim for that group is to filter research findings on Parkinson's to people with the disease. I have regular contact with people with Parkinson's, either directly or indirectly, via local groups, online conferences, and email newsletters. It was interesting to discover how Parkinson's presents itself with different individuals. The years of diagnosis do not define the severity of the condition in people with Parkinson's, as it is dependent on how aware the person is about their symptoms, and also how soon they seek help from specialists. I have encountered individuals who were transfixed in the stage of denial, even after diagnosis, and who are fearful and unwilling to be motivated to take on lifestyle changes. Most recently, my involvement with the charity has extended to further afield. For 5 years, I have been a patient advisor for UCB Pharma on their patient-reported outcome measures project. I am also involved in the University College London's research project on accelerating clinical trials using the multi-arm and multi-stage design. Earlier on this year, I attended meetings with representatives from Michael J. Fox Foundation on their new PD staging method based on the discovery of new biomarkers. Finally, a little bit more about myself. Since my diagnosis, I have held back on starting dopamine replacement, which is a personal choice. Instead, I am on an MAOIB inhibitor and a dopamine agonist, which I hope will help to preserve my brain cells for a while longer. I strongly believe in taking control of my well-being by putting into practice what research on Parkinson's has instigated. This means regular exercise of different intensity and varieties. For me, I enjoy yoga, swimming, and dancing. I also believe in the promotion of gut health by regular intake of kefir and a healthy diet of mostly organic produce. Recently, I have returned to part-time work, providing respite for carers of people with neurodegenerative disorders. I find this most stimulating, not only for my mind, but also for my spirit. That's all about me. Back over to you, Thilo.
Wow, that was amazing. Thank you, Carol. That was just inspirational, and thank you. Okay, so let's start from the beginning. Halal, what is alpha-synuclein? What does it do in health, if you can answer that, and why does it aggregate in diseases?
Okay, that's
tough question. Sorry.
So it's a very interesting question because very often when we're asked about what synuclein does, it's very rare that we're asked that what it is. And that's because usually there is an assumption that we actually know what is alpha-synuclein. So let me start a little bit with saying, what do we know about the normal synuclein before I talk about what we know about its function? Well, it's a protein that is highly abundant in the brain. It's expressed in different cell types in different parts of the brain, but also in other peripheral organs. It's generally enriched in presynaptic terminals. What we know very little about how it looks like in its natural environment in terms of the native protein inside neurons or other cells, or how many functional forms of this protein exist in the brain. It's a very highly dynamic protein that usually cycles between the cytosol and the membrane-bound state. And many of the mutations that are associated with the early onset form of Parkinson's disease are usually found in the part of the protein that regulates its interaction with membrane. I'd like to emphasize that most of our understanding about the structural and biochemical properties of synuclein is actually based on studying this protein in a test tube and not in the brain or the cell. And that is because it remains very challenging, if not impossible today, to be able to selectively monitor the shape and behavior of this protein in the presence of thousands of other proteins in the cell. So for example, and this may be relevant later, we don't know what the conformation of the endogenous proteins or what oligomeric form of these proteins look like. If you look at most scientific papers on alpha-synuclein, you always see this helical conformation of alpha-synuclein bound to the membrane. Yet this is not based on data on biological membrane. No one has actually determined the structure of conformation of synuclein on biological membranes. What we know and what we understand, that is, very much more about the abnormal disease-associated forms of the protein. We know that these mutations or increased expression of synuclein is sufficient to trigger Parkinson's disease. We know that the presence and detection of synuclein in the CSF in individuals is sufficient to enable differentiating Parkinson's disease patients from healthy controls with greater than 95% accuracy. We know that the presence of aggregated synuclein is one of the defining hallmarks of PD, but in some subtypes of Parkinson's disease, neuronal loss is observed in the absence of synuclein aggregates, and the presence of aggregates does not always lead to cell loss. The aggregated form of synuclein are also found in healthy individuals, aging individuals. Some people have argued that since aggregated form of the protein exists in both healthy and diseased brain, this suggests that synuclein aggregation is not responsible for neurodegeneration. This argument is based on the untested assumption that alpha-synuclein aggregates in healthy and diseased brain or neurons are actually the same. And we have now data that show this is actually not the case. Furthermore, these arguments do not account for the possibility that some individuals may have more resilient brains that are capable of neutralizing or counteracting the toxic properties of these aggregates, as we're seeing now a number of example cases from the Alzheimer's field. In terms of function, synuclein is a multifunctional protein with many new functions identified, almost every few months now. The question is which of these functions, when impacted or disrupted, contributes to the pathophysiology and clinical symptoms of the disease. Several, but not all, synuclein function are related to its binding and localization on membrane. It's been, synuclein, its presynaptic location and presumed some association with synaptic vesicles have pointed researchers in the direction that led them to identify several putative functions, including a role in neurotransmitter release, synaptic vesicle release, synaptic plasticity, membrane fusion. At the synaptic terminal, synuclein is associated with fusion of lipid vesicles. It's also known to act to sense and influence membrane curvature. It also has been, proposed to function as a chaperone that binds to proteins and help them fold properly so that they don't aggregate. For example, the cysteine string protein alpha chaperone for the snare complex deficiency leads to neurodegeneration, and deletion of synuclein exasperates that, whereas overexpression of synuclein or reintroducing synuclein rescues the formation of these complex and degeneration. So there's now lots of data now points to an important role of synuclein in the immune system, and the fact that synuclein can influence the immune response, and conversely, the immune system can also impact alpha-synuclein structure and function. So we're getting roles also for that synuclein plays a role in response to viral and bacterial infections. And it has an important role in immune signaling, and also some synuclein peptides can act as antimicrobial peptide. So I think synuclein is not a protein of unknown function. It has many functions. The question we have to decipher is which of these functions is relevant to Parkinson's disease, if any.
Brilliant, thank you. Yeah, multifunctional protein is a really good description. And the innate immunity or the bacterial virus protection is something I personally work on. So Gennaro, I wanted to ask you from industry perspective, maybe can you give us an overview of the different routes that can be used to target synuclein in people? I know that you ran the monoclonal antibody trial, but can you give us an overview of all the different ways that companies are trying to target alpha-synuclein?
Sure. There are different approaches, right? They might be divided mostly in two. One is acting on the wild type, and the other one is acting on the pathogenic form, the aggregates, or whatever is in the middle between the two. So some approaches are trying to bind, like the monoclonal antibody you just mentioned, prasinezumab, and many others, right? They are able to bind aggregates in a selective manner. This is happening everywhere, right? It's infusion in the body, and then also go into the brain. We infuse a very, very high dose in order to make sure that even if the monoclonal antibody go only 0.3%, right, 0.1% into the brain, will be enough to bind the aggregates. However, we don't know how much aggregates we have, and we don't know, as Hilal was saying, what is the real role, what is happening with them at the extracellular level, right? The other approaches that are targeting aggregates are trying to inhibit the formation or try to dissolve them, right? There are a few molecules that are in development, small molecules that are supposed to basically reduce this aggregation process. However, it's very difficult to pinpoint to a specific biological test. As I said, the monoclonal antibodies bind to the aggregates. Now, what happened after that is also difficult to know, because there are studies showing that they go into microglia. There are studies that don't do that. There are also forms when we have a brain shuttle, right? Some companies are developing that too, so to make more brain penetrant. But at the same time, it's still unclear for us what the aggregates do and how the drug interfere with them. On the other hand, as I was saying before, there are approaches that are trying to reduce the wild type amount of synuclein with the what's so-called antisense oligonucleotide, ASOs, and they are currently in development in a typical form of Parkinson's disease, more aggressive form, because this is an invasive procedure that needs to be done by intrathecal injection. And of course, when you target the wild type, reducing the production of synuclein is also possible to go after that with gene therapies, right? And there are a few companies working on this as well, trying to go after the production. Now, why this could work? Because if you have a patient where there is more production of synuclein, and we see that with the aging, right? The older you are, higher is the probability to have higher level of synuclein in the cytosol. And if this is pathogenic or not, we don't know, but at least we know that age is also associated with the high risk of Parkinson's disease. There are also evidence associated with different type of toxin, right, like pesticide, etc., that can induce an increase of alpha-synuclein that is supposed to be temporary, but it might not go to normal. And then if you have this increase, then you might be a high risk to develop Parkinson later down the line. Or maybe you are on the edge, right? And then this gives the final boost. So on one end, if theoretically the reduction of wild type might reduce the normal function of synuclein and can be potentially toxic, it could also be positive because we'll switch the equation towards having less probability either to form aggregates or to develop Parkinson's disease.
Great. Can I ask you a direct question about your trial? So I know you're not going to be able to comment on other trials, but the Pasadena trial and the Spark trial failed to meet their primary outcomes, their endpoints. What is the next stage? So there's been some reviews discussing these trials. What do you see as the next steps for monoclonal antibodies from your perspective?
I think it's a very exciting time because different monoclonal antibodies are currently tested, right? And this is scratching the surface. I know there are some people thinking, okay, we are at the end of this journey, but we are actually at the beginning. These are the first two monoclonal antibodies. There are many others in development. They are really different. Maybe some of them are more potent than others, and this could, by its own, justify a different effect. To respond to your question about the primary endpoint and what we are doing at the moment we have a big trial running called Padova. That is the second study that we started after the Pasadena results. And why we started this study? Because we saw, and I shared this in the New England Journal paper, right, on Pasadena and also in several congress, we saw that even if the primary endpoint was not met, what we saw was an effect on the MDS-UPDRS part 3. That is the majority of the component of the primary endpoint, and is what are the motor signs, basically slowness, tremor, stiffness, that are rated by the doctors. So the doctor rate the patient, and then if you look at that endpoint, we see approximately 25% reduction in progression over 1 year. And this is why we said, maybe the drug is working. It could be by chance, right? We cannot exclude that. But it could also be that this endpoint specifically is the one that can move over 1 year. And then we're testing this endpoint in a second trial that will give us the proof if this is actually the case or was just a chance finding. What we do also in the current trial, we do the study not only on treatment naive, that are people just diagnosed and treated with morbidity, what we did before. We also test the drug on top of levodopa because we want to make sure that the effect that we saw, it needs to be true and it needs to be meaningful for patients because we want to give this drug on top of the best standard of care that people can get.
Yeah, great, thank you. Before we get to Carol, I'm going to have Halal bring in a comment, but one comment, sorry. One instruction I've been asked to give is if you have a question, please put it in the Q&A. Some people are putting up their hand. We can't unmute you to speak. So if you have a question, please put it in the Q&A. Halal.
Yeah, I think on the issue of antibodies, I think we need to remember a few things. One is that most antibodies will target mostly extracellular synuclein. So this is synuclein that's probably responsible for propagating the pathology. Yet, and if you have cell autonomous aggregation, that will not be managed by the antibodies. So the main engines and machinery for producing aggregates is still active inside the cell. Another issue that I see with antibodies is most of the antibodies that have been developed are actually do not account for the diversity of pathology in terms of extensive, in the brain in terms of post-translational modification, or account for the fact that there could be different fibril strains. So if it turns out to be the case that, the differences at the level of the synuclein aggregates could account for clinical heterogeneity or inter-individual variations, then it's going to be challenging to develop an antibody for each one. So in my opinion, when it comes to developing therapies, because when a person is already diagnosed, you already have aggregates in the brain. So you're going to have to do two things. You're going to have to turn off the faucets so no more aggregation takes place, and then clean the aggregates are there. The antibodies are going to be useful, but we need alternative strategies that target the de novo aggregation and prevent aggregation from happening in the first place. One therapeutic approach is that has not been discussed, which we were successful in work that we did a long time ago with Jeff Kelly and transthyretin, is the best therapy, the best combination therapy would be a drug that stabilizes normal synuclein and prevents its misfolding in the first place. And then a combination of an antibody that targets specific, the different aggregated form of the protein. So I think we need to begin because, to accept that we will need some sort of a combination approach that target these different processes along the aggregation pathway of alpha-synuclein.
Yeah, well said. I think this combination idea is very good. And Gennaro is absolutely correct. This is the beginning of work with antibodies. You can't expect the very first trials to be successful, but you're learning lots. So Carol, what should the priorities be of industry for these clinical trials for Parkinson's? And I wanted to get your thoughts on the different routes of delivering these medicines. So Gennaro mentioned intrathecal, which will be, I think, is a spinal cord injection, intravenous, which the antibodies or pills. So what are your thoughts on as a person that could be taking up some of these therapies?
Sure. First, the amazing explanation by the two gentlemen on the science behind it is very enlightening. I think from the patient's perspective, we need to examine the different options in tackling this neurodegenerative condition. I think firstly, right at the beginning of the spectrum, it's important to realize that one of the options is to prevent the onset by tackling the causative factor. And whether that is inhaling or ingesting obnoxious molecules, or as Halal has said, that there are certain changes to the aggregation that can be stimulated by the reaction to the immunity system. Another option in tackling the condition is to lessen the impact of the disease by looking at how we can cope with the symptoms, or even tackling the side effects of drugs, because that can improve the quality of life for us as individuals with Parkinson's and maybe extend our lifespan. Then there's also the possibility of halting the disease progression, which is something that we always hope for. And lastly, which is something quite impossible, but it might happen, is to reverse the disease. So keeping those different options in mind, I will then answer your question, Thilo. What are the patient's priority? I think most importantly, from my point of view, which I have sort of said, implied in my introduction, that people with Parkinson's need to be aware of research findings. They need to be able to read the research findings, to go on webinars, and also to be in touch with knowledgeable others so that they know what is out there, because things are moving so fast these days. And by being aware of research, they not only can help themselves, but they can help others in the Parkinson's community. Other priorities would be, after understanding what the research findings are, is to motivate oneself to make those lifestyle changes based on the findings. And also, finally, but probably the most important, to partake in research projects, either as participants or advisors to research project. And I just want to quote a quote from Goethe, who is a writer, a German writer and a philosopher. Knowing is not enough. We must apply. Willing is not enough. We must do. So that is the important thing, the message I want to give out to all the people in the Parkinson's community. We must grab it by the horns and tackle it face on. In regards to what you said about using the lumbar puncture technique to extract the CSF, I have spoken to quite a few people with Parkinson's about that. There is always a jeopardization in regards to invasive procedures, especially those that had bad experiences. So the most important thing is demystification. So they need to be reassured that the procedure is fine, it's not going to hurt, it's not going to introduce infection. And maybe further down the line, there could be other means of extracting and measuring the alpha-synuclein aggregates, maybe via the blood, maybe via the saliva, maybe via tissue samples. So there's always hope for future work on this.
Brilliant, outstanding. Thank you. This segues very nicely into a question I have for Gennaro about measuring alpha-synuclein. So during clinical trials, either ones that have passed or upcoming, are you going to directly measure alpha-synuclein in a body fluid, and how are you going to do that?
We tried. We developed a skin biopsy assay, right? Unfortunately, the assay did not give the results as expected because it's difficult to find synuclein, phosphosynuclein pathogenic, right? Because the number of terminals, some people have tried with more success, but at least in Pasadena, and this was only peripheral synuclein, right? We also have CSF, and unfortunately, we cannot measure synuclein in the brain or aggregates in the brain yet. There are attempts on PET tracers, and it seems that finally we get some lights from a PET tracer that is currently going into humans in 2023, hopefully, from Merck. They got an award from the Michael J. Fox, the Griffin Award. So we really hope that this one, there are other companies. We also have PET tracers ourselves, and other companies have that. But the difficult part is to really have a PET tracer or a biomarker specific of the aggregates, right? And then we can measure monomers, we can measure total synuclein, but does not give you the same specificity that we need for our trials, because we want to use the biomarker to confirm the target engagement of the drug. I mean, the news, what is on the news and everyone is talking about is this seeding assay, right? That is around for a while and can define the presence of aggregate in the CSF, potentially also in other skin biopsy or other biopsies, but is at the moment not quantitative. So it can tell you if you have it or not, but we cannot use to test the efficacy of the drug. So this will be very, very important, especially to define the dose and to define which drug is working the most, right? For patients, it would be important to take a drug that works the most on the pathology.
Yeah. Great. Hello, what are your thoughts on quantitatively measuring alpha-synuclein from a person?
Yeah, we've tried a lot. So, we tried to look in the CSF for different modified form of alpha-synuclein, and actually we cannot find very much. I think phospho-S129, I believe the Roche group and other groups have also failed to be able to measure it as a reliable marker. We can measure total synuclein, but total synuclein by itself is not, even though we see a trend of a reduction, you end up with quite a lot of overlap between the cohorts, with usually about 40% of people with PD, without PD, having the same levels. It seems right now, one of the most promising is these RT-quick amplification assays. I think, well, we just published a review on this recently, and what we found is when it came to biomarker discovery, a lot of the work, has been done mainly on CSF. So we have not really systematically explored biomarkers, synuclein biomarkers in the blood. And I think this is something that we should do, as any future biomarker would likely to be the most useful one to be a blood-based. I think synuclein is going to be a useful biomarker. It in itself is not going to be sufficient for accurate diagnosis or to monitor disease progression. We're going to need to combine this with other biomarkers of neurodegeneration. The RT-quick assay is very promising, but I also share the concern that I have doubts that it's going to be quantitative and useful for monitoring disease progression. It's also not so far not useful now for early diagnosis, except in some cases, people with the REM sleep disorder. But it's going to be, if the day, it has two things that I think could add very much. The day we have an imaging agent, we will be able to see whether we can correlate seeding activity to the presence of synuclein pathology in the brain. And I think that will be big. That being said, I think there are a couple things about this RT-QuIC assays that we still don't understand. And one, we have yet to be able, if synuclein aggregates in the CSF, then we have to be able, when we amplify, to produce structures that look like what's in the brain. And nobody has been able to do this. The second important thing about this assay that we still need to work out is you can deplete 90% of synuclein from the CSF and you still get a seeding activity. So we still, cannot rule out that it could be synuclein or synuclein plus another factor that contributes to this. And I think, the other thing is, we assume most assays for RT-QuIC are based on using one substrate of synuclein. Yet, if the pathology is different in different individuals, that single substrate may not work for in different synucleinopathies, and therefore allow you to separate PD from other synucleinopathies. But I think, combining synuclein biomarkers with other neurodegeneration biomarkers will probably be the way to go. And having an imaging agent will most likely be transformative with these biomarkers.
Great, thank you. We're going to go to a few of the questions that are appearing in the Q&A. First one I'm going to mention is, where are we in terms of gut-mediated alpha-synuclein aggregation? Can this be used for early diagnosis? And will it be part of combination therapy? Meaning, should we also target alpha-synuclein in the gut? Maybe Hala?
Yeah, I think this is one of the, the exciting development over the years, the past recent years, this gut-brain axis, also the role of viral infection and in initiating pathology, because it sort of gives us an opportunity for early intervention, especially given the tight association with GI, dysfunction in some Parkinson disease patients. If we can couple this with biomarkers, then that would give us a huge opportunity for early intervention. The challenge I see there is when it comes to understanding synuclein in peripheral tissues, we're way, way behind where we are in the brain. There is an assumption that, that synuclein pathology in the gut is similar to the brain. We have not done that. We don't know if this is the case. And therefore, if they could be both targeted, using the same antibodies or approaches. Most characterization of synuclein pathology in gut and peripheral tissues is actually very superficial, relying mainly on phospho-S129 signal, and very often without establishing that it's really pathological synuclein. So I think, it's in terms of therapeutic opportunities and window, it's a very exciting development, and I think but there is still a lot of work to, investigate the link between brain pathology and gut pathology, and whether both can be targeted using the same approach.
Great. Gennaro, here's a question for you, tough one. In case one of the main issues about alpha-synuclein aggregation preventing the normal function of alpha-synuclein, is it possible to increase the levels of alpha-synuclein to see if this helps against a background of aggregated alpha-synuclein? So is increasing alpha-synuclein a way forward, essentially, what this question is asking?
I think this is a question that, the biology, Hilal was explaining the biology in the very beginning, right? Doesn't look like that this approach might help, right? At least of what we know so far. Maybe Hilal, you can expand on that, right? I think one element is we have aggregates, right? But while we have the aggregates, maybe there is something that happened that made more production of synuclein at the level that synuclein cannot stay in the monomeric form, and then it starts aggregating because there is too much. And there are biological evidence of that. If you have duplication or triplication on the wild type copy of synuclein, then you start having more aggregates, and then you get disease earlier, right? Even young age, 40 years old, 30 years old, right? As more synuclein you have, genetically wild type synuclein. So people might argue, okay, but if you restore the normal function of synuclein, or you harm that, you might get worse. And it's true. But I think the aggregates are kind of a way for the brain to respond to the excess of synuclein, right? So by themselves, they might not be toxic. But if you remove them, they're kind of a sink. So where there is a lot of synuclein that shouldn't be there. So if you remove them, you let the body to create more aggregates that take away the excess of wild type synuclein that is there and having a negative function. Think about a U shape, right? In the big, if you have too little synuclein, it's not good. If you have enough synuclein, it's good. But if you have too much synuclein, it's gonna be detrimental. And this is where I think both antibody and reducing the wild type can both work in combination, as Hilal was saying, eventually stabilizing. I really like this idea of stabilizing the wild type that will not go into aggregates, right? So you have more pushing the curve to the normal function. But bringing more synuclein into a brain that already have aggregates, I think might make is a really bad idea. In my view, at least. I could be wrong.
Yeah. It sounds dangerous to me. It's related to the next question. I'm going to let Hilal jump in, but it's a very similar question. How can both the inhibition of alpha-synuclein and overexpression of alpha-synuclein have advocates in the community? So, and as we said, the genetics tells us that too much synuclein, either duplication or triplication, aggressively increases Parkinson's disease, and triplication being even more severe. Hilal.
Yeah, I mean, I was going to say that, first, I think it's very, synuclein is a protein with a function, right? So we need to accept the fact that we don't have to choose between a loss of function and a gain of toxic function. It's likely that both of these contribute differently at different stages of the disease, And loss of function is also not restricted to synuclein, because as aggregates are forming, they could sequester other functional proteins. The second misconception, I think, is always that if you have synuclein aggregation, you're always depleting the monomer. That's actually not the case. Neurons are able to adapt. We have done these experiments. We can see aggregation for up to 14 days without loss of synuclein monomer. The only time we start to see synuclein monomers is when we see the conversion to Lewy bodies and cell death, because then it's even more dangerous. So the neuron can compensate for synuclein level. Now, one of the most consistent factor in terms of promoting synuclein pathology is protein levels, whether it's viral infection, whether it's duplication, whether it's genetic polymorphism. You increase the level of synuclein, you increase pathology. Now, if you already have seeds, it's like, that's even worse in terms of the application. So we've actually argued quite a lot about this, that, a few months ago, we decided in the lab, we do this experiment, and we did it. So what we did is we initiated synuclein aggregation in neurons, and we, up to 14 days in the seeding model, which we published a few years ago, and was developed originally by Virginia Lee and colleagues, we can see synuclein aggregates and pathology without major cell death. You see some synaptic dysfunction and some abnormalities. And then we asked, what happens if we bring synuclein back? And what happened is two things. We got increased pathology and increased cell death, right? Because you add more synuclein, you have seed, you have pathology. Now, what the experiment that we are trying to do now is to say, if we go under conditions where we begin to see cell death, can we, you would have to bring in a form of synuclein that does not aggregate. And now we have engineered sort of a monomeric form of synuclein. And the question is, if you bring this form of synuclein that doesn't aggregate, can it rescue or prevent, cell death in the presence of aggregate? That's an experiment we have to do. What I can tell you is that in order to engineer a form of synuclein that doesn't aggregate, you have to introduce mutations in the region associated with membrane binding. So there is a risk that you could also, have something that has abnormal interaction. So I think all of these questions are very important, but I think where we need to focus our effort is in designing the right experiments to answer these questions instead of constantly arguing who's right and who's wrong. You know, that's where our energy should be, is what would be the experiment? If somebody argues for loss of function or a gain, what would be the experiment? And there is a large number of people in the community, we can do it and then let the results speak for itself.
Spoken like a true experimentalist. Yeah, I completely agree with that. Let the data speak for itself and back up what your claims are with data. But let's get back to people in clinical trials. So Carol, can we get your thoughts on how clinical trials are designed, how you and UCB are maybe working together? And can you give, I don't know if you know the clinical trial landscape for targeting alpha-synuclein at the moment, but if you can comment on these, that would be brilliant.
Yeah, well, I can only comment on it as far as I am a patient rather than a scientist, but I'm a realist and a pragmatist. And I think we need to think of clinical trials and how it is designed by looking at resources and priorities. And those two areas are vitally important because in regards to resources, we're talking about funds, we're talking about manpower, and we're talking about time. And those things need to be considered when designing clinical trials. And I will use that to illustrate it when I talk about the ACT-PD and the UCB outcome measure project. In regards to priorities, of course, patients has got their own unmet needs and their wishes, but this is also in competition with societal needs and healthcare system burden. So when regulators like the MHRA and NICE look at whether clinical trials should be conducted or not, they need to consider those factors. It's not just what the patient wants, it's what society wants and whether healthcare systems can cope with that. So back to your question about trial design. The accelerated clinical trial, the ACT-PD project I'm with at the moment with UCL, I'm going to use that as an example first. They have considered the need to look at timing, because the average clinical trial at the moment to get a drug onto the market and into the patient's mouth, putting it crudely, takes up to 10 years. So the accelerated clinical trial project is looking at the multiple arm and multiple stage method. They are trialing drugs, in this case, 3 drugs in parallel with one another, alongside a placebo arm. So in other words, they're going to try and speed up the testing of the drug and hopefully cut the timing of the drug fruition, coming into fruition, down to about 5 years. So in regards to how the people with Parkinson's are involved in this project, we are very much involved in the planning of the trial, right from the beginning, looking at how we can impact on the infrastructure, on the resource, on the treatment selection and outcome measures. So it's vitally important to have the person with Parkinson's to be there in the center of discussion. As regards to the UCB outcome measure project that I was involved in for 5 years, they are looking at patient-reported outcomes. So that is very much something that I'm very interested in, because it's using quantitative and qualitative data. So the qualitative data collected from patients' lived experiences are used to match against quantitative measures. So this need for triangulation is a very interesting movement. So what Halal and Gennaro has said in regards to the scientist perspective, using objective findings can be cross-validated with qualitative findings from patients. And in regards to recruiting patients in the early stages, at the moment, we're relying on inclusion criteria that looks at the number of years the patient has been diagnosed. And that is an extremely flawed method, because as you well know, the Hohen and Year scale doesn't actually reflect truly on what stage the patient's disease trajectory is in. So the new biomarkers that Halal and Gennaro has mentioned, whether it is using a CSF measurement or blood or saliva, etc., can be extremely useful to allocate patients into the right part of the trial. Because when you say early stage PD, it doesn't mean anything if you're just recruiting patients under 5 years of diagnosis, because it all depends on when the patient go to see their specialist.
Great, perfect. We've only got 9 minutes left, so I'm going to ask Halal to chip in. But just before I do, I just wanted to say, if you're looking for clinical trials, Michael J. Fox Trial Finder, PD Trial Tracker are two really great sites. And also the Cure Parkinson's Link Clinical Trials is also a great place. So the Ambroxol trial that some of you may have heard about is currently recruiting. And this is a trial as a drug that is meant to target and reduce alpha-synuclein. So Halal, what are your thoughts on clinical trials and interaction with industry?
Something on the clinical trials, because the failure of drugs in clinical trials have huge implications, both for the science, for the patients, and for the community. And it is my opinion that in many times drugs are advanced to clinical trial prematurely. And I have reviewed data of many compounds, of many things that, based on the data published, should have never made it to clinical trial. So I think we need there some sort of a step for reproducibility or validating the science before we invest millions and millions of dollars and subject patients to these things, so that at least the main scientific foundation for taking something to a clinical trial should be validated. Yeah, now, we often hear from industry that academia, the large percentage of the data is not reproducible or not, but we never have, the same transparency with industry. The second important thing, I think, with industry, what I would love to see is publication of negative data. The industry are very good at jumping to benchmark their technology against others. And very often they would identify that a drug that is going to clinical trial or in clinical trial is not working, or is not working as advertised, but that data never makes it, And finally, industry relies heavily in science generated in academia, in tools generated in academia, but we get very little back in terms of science and tools from industry. They're very good at developing methods, assays, and tools, but everything ends up in the -80. And I think we really need, if we all care about advancing the disease in patient, we need to unlock some of these fridge and begin to share and give back to the community for the sake of improving reproducibility, transparency, and accelerating translational research.
Gennaro, come in, please.
I think that's a great call for action, right? And I can say we are working with, I was in academia before industry, right? So of course I'm biased, but I work with several academics trying to do exactly what Hilal is saying. And I encourage any of you listening today, if you have ideas, if you want to test something on Pasadena, on trials that we've conducted, we have samples, as Hilal said. They don't need to stay in the -80. If they are a good idea, we collaborate. We collaborate at least with 3 groups at the moment. Spontaneous collaboration, this is what we want, because the idea needs to be, my dream, as I said before in the beginning, is to make things to the patient. But in order to do that, we need to speed up, and also what Scarlett was saying, the time from the idea to delivery. there is too much in the middle. We need to streamline, we need to have pre-competitive consortia, we need to think together what is the right thing to do and stop compound early, as Hilal was saying before, that don't have the clarity, they don't have target engagement, they don't have preclinical data, they don't need to go anywhere. They should be stopped. And then it's difficult sometimes. I can tell you, I've been on the other side now. And when you work on a program, there are a lot of people working. It's difficult to say to everyone, look, now we stop, right? Everyone has a hope because maybe they have patients, they are themselves patients, they have family members. They want the drug, right? Even people that are in industry themselves, right? Don't take it as just, there are a lot of, ideas about industry, but I can tell you, many people are really motivated for personal reason everywhere. And it's difficult to admit, okay, this drug is not working now, but we have to do it. So we make space for drugs that eventually will work.
Can you answer this question? What lessons can be learned from clinical trials in Alzheimer's that target tau and amyloid, Gennaro?
What we learn, especially in the last period, that you need to have very potent drugs before you can put to bed the theory. Until 9 months ago, I would say, everyone here would say maybe amyloid is wrong, right? Why people are still pursuing amyloid? I would bet that. And now we have two drugs that have clear data, right? Of course, the effect is not extraordinary because it's only one of the cause of progression of disease. So what we learned for me is that we need to invest heavily on the drug, make sure that the drug works very well before we test them in large clinical trials, and not be pushed by, okay, we need to be fast. I'm contradicting myself because I've said before, I said we need to be fast, right? But we need to be fast with the right drugs and not just fast to run into the failure, right? And stop early. If you look at the amyloid trials that failed, for example, in Alzheimer's, they did not have strong phase 2 data. They did not have strong preclinical proof of concept that have the drug that actually worked. Quickly removal of pathogenic form of amyloid seems to be the way to go, and full removal, right? We had drug in our company that did not meet that criteria, and unfortunately did not work at the end because it did not fully remove quickly the pathogenic form. And this is why I think we need to invest heavily now. If we have proof of concept with even the next trial that we're running with monoclonal antibody for aggregated alpha-synuclein, is that just scratching the surface. We need very potent drug to do this quickly and fully.
One more minute for Hilal, and then I'll wrap up.
If I just add briefly, I think the other thing we've learned is going to take a combination therapy. We have multiple pathways that are at play. You're going to need a combination therapy, in order to tackle these diseases, and we need to figure out smart way of designing clinical trials, where we introduce combination therapy that maybe even take into account personalized approaches. I think the other thing I wanted to say about, well, I lost my thought, so I think it's good.
Okay, it's okay. We're going to run out of time shortly. So first of all, I just wanted to thank all three of you for some really, really brilliant contributions. I think some take-home messages are alpha-synuclein is a very good target, and we need to keep working at it. Failed early clinical trials doesn't mean anything. We need to keep going at it. I think increased academia industry collaboration is a huge missed opportunity here. I think there's lots of tools generated in industry that academia can use, and industry working more closely is with academia is important. So Hilal's point about strong proof of concept, strong data to go to clinical trial is so important. I think I've seen it as well that that clinical trials happen well before they should happen. So that sounds like it should slow things down, but in the end, it will give us better results. And finally, no magic bullet, it sounds like. So combination therapy is going to be probably the answer to slowing and stopping Parkinson's disease. And I think the patient selection, so Carol's message about choosing the right patients at the right stage is very important. In years after diagnosis is certainly not good enough. So much more personalized approach to selecting the people for clinical trials, that could include genetics as well, is going to be the key. Okay, thank you very much, everyone. Please send a message about this webinar. It's going to be recorded and online, and we will see you next time. Thank you.
Thank you very much.
Thank
Thanks
you.
for having us.
Yeah.